Chapter 210, The Origin of Life. The challenge presented in this chapter may be assessed from the following. Russian biologist Kirill Yeskov, in his book The History of Earth and Life on It, states that many leading biological journals, for example, the Journal of General Biology of the Russian Academy of Sciences, do not accept articles on the origin of life, do not accept in principle, sort of like the projects of a perpetuum mobile. Evolutionary scientists are trying to avoid this issue. Darwin, and the vast majority of his followers, talked about evolution not from the very beginning, but taking the existence of unicellular organisms for granted. Darwin believed that the cell is something primitive, a kind of ball filled with a certain liquid. For him, the origin of the cell was not a problem of particular importance. Well, it just appears by itself as the bubbles on the swamps. We know that even a nuclear-free cell of prokaryotes has a complex structure, and the global mind has worked many million years for its creation. If we can understand how global mind came to this revolutionary solution, the description of the further evolution will be a less difficult task. Could it originate by accidental coincidence or a combination of elementary chemical elements and compounds? The famous American biologist Stuart Kauffman, like many other researchers, calculated that the spontaneous accidental nucleation of a protein molecule is characterized by a probability of 10 to the negative 20 to 10 to the negative 40,000. The fact that a single-celled organism, such as a bacterium, possesses a certain intelligence does not cause doubts among people familiar with the subject. Some microbiologists consider the level of this intelligence to be worthy of respect, and the mind of the colonies amazes even the researchers who observe them. A group of Israeli scientists conducted an observation on the behavior of the bacteria colonies in the search for food. To do this, they placed the nutrients in islands of different sizes and tried to predict the trajectory of the colony's movement, that is, the multi-billion army of the simplest bacteria, to the goal. Usually, the colony moved towards the food, guided by the growing chemical traces of its presence. The closer to food, the more chemical traces became tangible. The colony, like an army commanded by an experienced commander, sent out scout groups that were probing the possible directions and sending the results of their observations back to the main group. After that, the column marched straight to the target and attacked it. In one of the experiments, cunning researchers divided the nutrient into 3 islands and tried to predict the behavior of the colony. The most attractive and large pile of nutrients was located in the distance. A bit closer was placed a portion of a slightly smaller size, and on the sides of it were placed quite tiny piles. Scientists watched which way the colony would choose, go to a distant large island or first capture the second largest. The results stunned them. On the way to the second largest island, the colony unexpectedly pulled out 2 lateral wings that captured the smallest islets of food. The colony center, slightly changing the trajectory, rushed to the second largest island. After eating the first 3, all the groups of bacteria again merged into a single stream, rushing to the furthest but the biggest and most desired goal. None of the observers with scientific degrees could have predicted such tactics. Scientists would do it differently, and it turned out that bacteria colony path was the most reasonable and optimal. It seems obvious that bacterium, as well as any other living creature, could only be created by a more intelligent entity than itself. How then could the first cell appear in the world? Who produced it? It can be seen that the bacteria colony has an incomparably higher intelligence than a single bacterium, or even a group of PhDs. Consequently, the colony of bacteria has a different quality than a single unicellular structure and can, in principle, create a new species. Ben Jacob observed the formation of a bacteriospore, which he considered as a new species. Let us recall that in order to achieve a certain level of intelligence, a computer must have a critical quantity of its constituent cells, transistors. The question is, could the first cell originate through combinations of more elementary formations? Could life, as such, have been the result of intellectual activity of prebiotic but complex chemical formations? Even the supercomputer's transistor does not have the intellect or free will. It is not alive. Let us imagine what qualities such a chemical formation should have. First, it must self-germinate in certain natural conditions. Secondly, it must duplicate and multiply. Thirdly, this chemical compound should contain a certain amount of information. It must also disintegrate. For what kind of life is it that is not capable of dying? Finally, the disintegrating components must transmit all or a part of the information stored in them to the newly born components. The question of such chemical compounds' existence and the possibility of their spontaneous generation is a fundamental one. Surprisingly, such compounds are known. They exist and are well studied. Let's turn to the fundamental 3-volume of Manfred Eigen and Peter Schuster work, The Hypercycle, a principle of natural self-organization, and try to understand this extremely complex and specialized study. The authors argue that RNA molecules, unlike DNA, are capable of self-assembly under laboratory conditions. Thus, the RNA molecule is capable of self-copying. This gives hope for the emergence of life on the basis of RNA, the notorious RNA world. Let us recall what RNA is and why its properties are necessary and sufficient for the existence of life in general and intelligent life in particular. As one can recall, RNA, ribonucleic acid, is a single helix with numerous links called nucleotides. RNA, like DNA, carries information encoded in 3 neighboring nucleotides, codons. The sequence of nucleotides allows RNA to encode and store genetic information. DNA also carries and transmits genetic information by the codons. However, the double helix of DNA needs to be unfastened to get access to the stored information, while RNA has an open structure with immediate access to it. RNA is capable of autocatalysis, that is, self-reproduction, and even a reproduction with modification. The daughter RNA may be different from the mother molecule. The phenomenon of RNA autocatalysis was discovered relatively recently, in the 1980s, by a group of scientists led by Thomas Cech. RNA is able to create unlimited colonies even without being compartmentalized, i.e., being enclosed in the membrane. Alexander Chetverin, head of the Laboratory of Viral RNA Biochemistry at the Institute of Protein of the Russian Academy of Sciences, and his colleagues discovered and patented molecular colonies called nanocolonies. Instead of compartmentalization, RNA colonies use porous materials. Chetverin believes that the size of such colonies is limited only by the physical dimensions of the porous material on which they are located. Thus, RNA colonies could exist and multiply, duplicate, even before the invention of membranes. Chetverin suggests that further compartmentalization with the formation of the membrane could occur later by forming a lipid film surrounding the RNA molecules. Summing up, A, the complex molecules replicate and multiply, and B, the broken molecules do not lose information but transmit it to new molecules. The general information contained in the colony increases despite the death of individual RNA molecules. This is almost life. American microbiologist Carl Woese, analyzing various data, concluded that the first organism was not a cell. He called it progenote, or a proto-organism, very similar to the Solaris of the RNA community. The idea put forward by the Russian academician Spirin, that is, a community of macromolecular complexes that can reproduce themselves. They can exchange genetic information. According to Woese, the cells originated from such a community. The transition to modern cells, the Darwinian threshold, occurred when organisms evolved translation mechanisms with modern levels of fidelity. Improved performance allowed cellular organization to reach a level of complexity and connectedness that made genes from other organisms much less able to displace an individual's own genes. Horizontal gene transfer during this era was responsible for the vast early evolution of complex biological structures. The idea of a Solaris RNA well explains how life could quickly arise. But can RNA appear by itself? Back in 1975, Manfred Sumper and Rüdiger Luce from M. Eigen's laboratory demonstrated that in a mixture with no RNA containing only nucleotides and Q replicase, a self-replicating RNA can arise arise spontaneously under certain conditions. In March 2015, scientists from NASA reported that complex DNA and RNA were obtained under conditions corresponding to outer space. They use a chemical compound contained in meteorites, pyrimidine. Just recently, in January 2018, NASA reported that the astronomers discovered complex organic molecules towards the N113 star formation region. The hypothesis of the RNA world is not yet fully proved, although it is very attractive for proponents of spontaneous generation of life. It is not the only possibility, however. The already mentioned American biologist and researcher of complex systems, Stuart Kauffman, in a 600-page book, The Origin of Order, discusses other versions in detail. Kauffman suggests that the simple and complex systems can show strong self-organization. This spontaneous order can be the subject of natural selection and random changes for the further selective design of random selection. Kauffman developed an unorthodox but verifiable theory of spontaneous crystallization of sequences of biochemical compounds, in which they catalyze the construction of new blocks. He suggests that the construction of polymer constructions on the basis of RNA can happen spontaneously. He proposed that life began with simple, self-replicating molecules and gradually learned the complexity. Summing up this chapter, it is possible to put forward the following assumption. In order for life to emerge, the accidental creation of complex components is not a prerequisite. A transistor is a very simple element. It has only two states. To obtain true life, that is, a powerful self-developing biocomputer, three conditions are necessary. 1. A set of a large number of elementary components that are capable of replication. 2. The stimulus for the development. 3. Initial impetus, which may be the inevitable imbalance of the system.